Thanks for coming. I really want to thank the organizers for inviting me. I can't overstate how interesting the conversations have been that I've been having just yesterday and this morning. I'm looking forward to the rest of the week of more of the same. Before I also want to make sure I thank, acknowledge my colleague that have done this work with me and interesting conversations. I've had some funding. Changying Lin was at Los Alamos until she was rudely expelled from that by some of the lawless forces that we are all aware of. But thankfully, and careful, they listen. But I'm very grateful that she did manage to get a position at Ohio State. So she's moved to Ohio State as soon as her visa comes through. Let's keep our fingers crossed for that. Okay, so I'm going to talk about phylodynamics today. And the way I think about phylodynamics is basically getting two things to talk to one another. On the one hand, we have phylogenetic or genealogical trees that we get somehow or other from genetic data or genomic data. And we want to, on the one hand, understand, so we have the trees, we also have mathematical models of one kind or another. Typically, these are stochastic processes. And we'd like to be able to understand, is this on?
This must be too much. Yeah.
That's the way it usually works.
No, but you don't want to.
Right.
You did that.
Okay.
That was the feedback. No, it was on.
Right. So mathematical models and trees, right? And so on the one hand, for any given mathematical model, we can understand no, we're not. There's something else going on. I'm going to stay away from there. We can attempt to understand what signatures those models leave on the shape, those generating processes leave on the shapes of trees. And then, of course, if we can solve the problem, the reverse problem, the inverse problem, we can try to extract information from the genomic data, from the tree structure, to parameterize and infer the structure of these models. So the work that I've done personally has been primarily in epidemiology. So the model that I've got here is a compartmental epidemiological model of a kind that'll be familiar, I hope, to some of you at least. And these kinds of models express heterogeneities in the progression of disease, the nature of hosts, their susceptibility, transmissibility, and the strain structure of the thing. The different compartments represent hosts of different statuses vis-a-vis infection. All right. Okay. Can be trusted. And importantly, some of these hosts are infected with a viral pathogen, right? And so it's on the basis of that, the genealogical relationships that emerge among the pathogen samples, that we hope to be able to infer something about the dynamics of these models. So it's I can ask, what would an ideal inference method look like, right? So crucially, it should allow us to ask at least the questions that we want to ask, to express the scientific hypotheses which in really many contexts involve demographic rates that vary in time, that depend in nonlinear ways on state variables, that are heterogeneous with respect to a bunch of different kinds of structuring variables, spatial location, host age, and so on and so forth, behavior. So we'd like to be able to ask those kinds of questions. We'd also like to be able to use the data as fully as possible. So we'd like to find methods that achieve maximal statistical efficiency if it's possible. On the other hand, we'd also like these things to be easy to use. We'd like the computations to run in under 4 seconds on your wristwatch. And we'd like them to be robust to violations of model assumptions. And of course, as my mother always told me, these are nice things. We can't have nice things, right? We can't have all of these things. It's impossible, right? Even just trying to satisfy the maximal efficiency and the robustness at the same time, there's a trade-off. And of course, the more complicated the models we have, the more complicated the data we have, the more computationally expensive it is. So as a result of this trade-off, which is of course well understood by all of us, I think, in the room, a lot of work has focused on the feasible, the computationally accessible approaches that make approximations of one kind or another that allow us to actually perform computations. The price we pay for that computational ease is in a mismatch between the questions we want to ask, the scientific hypotheses we want to propose, and the methods we use. So what I'm going to do today is explore what does it take to get methods that do this for us, putting aside for the time being the question of computational efficiency. Okay, so let's jump in. So to suppose we have some data in the set of genome sequences, we can imagine there's some kind of a genealogical tree or phylogenetic tree that relates those sequences. We have a sequence evolution model, some kind of nucleotide substitution model, amino acid substitution model. And then the thing that I've been yammering on about, a dynamic stochastic transmission model of some kind of a generating process that operates at the scale of a population. And we may have some other data as well, perhaps in the form of time series on the system. The likelihood then, it's easy to see, factorizes in this way. It can be expressed as an integral, where the integral is taken over the space of all possible genealogies. The thing that I've highlighted in blue is familiar to people who do work on phylogenetics. The phylogenetic likelihood, common choices there would be the same kinds of choices that Joe Felsenstein originally made. But the thing I want to focus on today is the thing in red, because it relates the phylogeny to the dynamics, we might call the phylodynamic likelihood. So people, of course, have been very interested in this problem for a long time, and there are a couple of widespread approaches to this, many of them based upon the Kingman coalescent and variations thereon. And these are approximations, these are exact in the case of very, very restrictive population assumptions about the population process. But they're good approximations when we have large populations, small sample fractions. And importantly, all of the analytical tractability here comes from the ability to work in reverse time. So you took those microphones away, but they're still here.
So I think people on Zoom are having trouble seeing the slides. Maybe
not. Is it not zooming right now?
Oh yeah, it's this slide share. Yeah, no, it's not. You're not sharing slides. I just found that out. Yeah.
Okay. So let's pause. Pause for a moment. And
Yeah. Okay, can we share on Zoom?
I think that's it.
How is it the microphone wasn't an issue before? Is it because I have a booming voice? Maybe I'll turn this down. But then the Zoom people won't like it. This does not have a volume control. It's an up arrow and a down arrow. Yeah, I have a hard time.
One problem solved.
Okay, and I'll just stand back here and I'll be calm. All right, so like I'm saying, the justification for the use of coalescent approaches in this kind of a problem relies upon a large population assumption, relies upon the assumption that the sample fraction is small. And similarly, for the linear birth-death process, it's the second major category of approaches that are at the basis of existing widespread phylodynamic inference approaches. These typically, in order to apply in the presence of nonlinear phenomena, rely upon large populations, small sample fraction assumptions as well. The reason these methods are available to us and are useful is because they're tractable, and that tractability stems from the ability to carry out certain arguments in reverse time. The justification, the details of that are different in the two different cases, but that's the key feature that works there. The problem, of course, is that most of the models that express scientific hypotheses of interest don't lend themselves to time reversal. So one can't reverse those expressions in any meaningful sense. So that's the nub of the problem. You solve that problem, but now I can't forward the. Maybe I just need to click on it. There we go. All right. Okay, so what I want to do quickly today is show you how a given population process, some kind of a Markovian process at the population scale, induces a genealogical process in a unique way. That is to say, a process on the space of genealogies. And then I'm going to derive the exact likelihood of an observed genealogy as a solution to a particular nonlinear filtering problem. Interestingly, all of the existing methodology that we have for phylodynamics they all fall out as special cases of this theory. And although the general problem still is a hard problem, I'm not making a hard problem into an easy problem. I'm only making a hard problem into a hard problem of a slightly different form. At least in some cases, the equation lends itself to calculations that are feasible, and I'll show you some of those. So to give you a sense of what I mean by a process at the population scale, I want to think about, these are just some examples drawn from epidemiological work. We have, the simplest one is just a model where there are two demes here because the virus, although it's present in both hosts here in this box and hosts in this box, they behave differently. They're infectious when they're in this box, but not when they're in this box. Here's a two-strain model that I showed you before where there are four demes because, these two are the same as this, these two are the same as that, but they differ in the strain that they contain. Here's a model where the hosts differ in terms of the symptomatic nature of their infections, with asymptomatic infections going down a different pathway than symptomatic infections. Here's a model where the heterogeneity in host behavior is captured by different kinds of, by putting the hosts with different low spreading behavior or high spreading behavior into different boxes. These are just a few to sort of fertilize your imagination about what's possible. The key thing about this population process from a mathematical point of view, it just simply has to be a non-explosive Markov jump process at the moment on an arbitrary or almost arbitrary state space X. And of course, such a process is characterized by two things. Its initial state distribution at some time, say 0, and a kernel of jump rate. So if the rate of jump from state X to state X prime at time t is this function alpha, this has to, of course, be a non-negative and sum to something finite. Those are the restrictions on the population process. So this is extremely broad. It can not only encompass all those small, low-dimensional models that I showed you before, but really anything up to countable dimensions. And the jumps in particular don't need to be simple. We don't need to assume that only one thing happens at any given time. One really nice way to compactly summarize all that, and that's useful in the math that comes a little bit later, is in terms of the Kolmogorov forward equation. And you can see that this is what looks like a partial differential equation, but it's really just a whole bunch of ordinary differential equations, one for each value of x, for some quantity y. And if y has that initial condition and satisfies the Kolmogorov forward equation, then just by integrating over any set, integrating that W density over any set, we get the probability of finding the system in that state, in that set at that time. Okay, so for those of you, well, it's maybe helpful to just point out that this Kolmogorov forward equation, in most cases of interest, and in all the cases that I showed you before, we have these integrals that actually will reduce to just sums, right? And in fact, sums with a very few number of terms, because they'll basically, these kernels will have delta functions in them. So in the case of this SEIR model, we have 5 different kinds of transitions that can happen. If we're in a state X here with some number of susceptibles, exposed, infected, and recovered individuals, then a transmission event changes the state in a particular way. A disease progression event where somebody moves from E to I changes it in a particular way, and so on with the other different kinds. We can group these things now into different categories. Okay, and in this case, there's 5 transitions in there. There's one in each of 5 different categories. These categories are distinguished by their impact on the lineage structure. So a transmission event is a branching of lineages when one lineage moves into a new host, so it branches. A progression event is a migration event where a lineage moves from one deme to another, in this case, from the E deme to the I deme. A recovery event is a death event in this case because that lineage is now gone extinct in that host. A waning event is an event that is neutral with respect to the genealogy. It only affects individuals that are uninfected, and so it has no impact on the genealogy. And then finally, a sample event introduces an actual genomic sample that'll, of course, go into the data. So we can, this becomes a marked jump process. We'll use U for the set of marks. And if we write this out, if we actually expand those kernels out in a way that makes sense for this particular model, it just becomes a sum, right? So we have an equation here that is not nearly so scary as it seemed with the integrals in place. But I just want you to keep that in mind, that these integrals give you mathematical generality, but in all specific cases, just reduced to something much simpler. Okay, so now I have to talk to you about what a genealogy is. So in order to make the proofs, we have to think very carefully about this. And of course, there's multiple ways of expressing what a genealogy is, or these kinds of tree-like structures. Intuitively, it's a tree-like structure that expresses the family relationships, the relationships of shared ancestry between individuals that are extant in a population, the black dots here, and possibly some samples genomes as well, indicated with the blue dots. But in order to really formalize this to make the proofs work, one has to do a little bit of work. There's nothing terribly important here. If we had more time, we could go into that. The key thing to recognize is that, as I'll say later, the genealogy can be represented as a function of time, right? As a time-dependent function. But of course, there's more structure to the genealogy in a multi-deme situation than I showed just there. Each one of those lineages at each point in time was by assumption, in one of the demes or others. And so we can think about that as being, just simply represent that using colors on the tree. Okay, and again, one has to think a little bit about that. Formally, a genealogy then just becomes a triple of a time, which is the sort of overall depth of the genealogy, a variable z, which is itself a function of time, which represents the topological structure, and the coloring structure represented by this variable y. All right, so that's how mathematically we're going to represent. I was being good. All right, maybe I don't know what to do. I'm just going to speak quietly. Yeah, there's probably more than one mic.
No, you're.
Yeah, it has to be more than one mic. There is a hidden Zoom.
Well, I turned off
Now we can hear.
a speaker. All right, we'll just keep turning things off until it all works, which is basically how the math works as well. But right, so as I said to you, the key ingredient here in the construction is that the Markov population process that we've assumed, right, that's the content of our scientific hypotheses, induces a process on the space of genealogies. And the way that's done is a fair amount of work in the paper on making that work. It's not terribly interesting, it's fairly finicky, but the key thing is that it allows us to characterize the probabilities of each individual genealogy given a particular history of the population process perfectly. And the key assumption that makes that all work is that lineages within a deme are exchangeable from one another. This is a funny thing. This is an additional mathematical assumption that one has to add, but it's very frequently not recognized as such, right? It's such a minimal assumption that's being added that it's essentially tantamount to the assumption that we're adding no additional structure to the population beyond the structure that we've assumed in the Markov process. All right. So you're going to have to take my word for it that this construction works to give us a random genealogy process for every realization of the population process. And that genealogy process, as I was saying before, represents the relationships amongst all the extant individuals in the population as well as the samples. But of course, we can't see that what we can see is what you can call a prune genealogy, where we throw away all the black dots. We throw away all the lineages which are ancestral to black dots, but not ancestral to samples. This results in a prune genealogy. The color remains here. And of course, we can't see that either. Well, before I go into that, I emphasize one more time that this can be represented as two functions of time, y and z. They're mathematically a little bit complex, but they're basically just functions of time. We can't see the color, of course, in any real or most real examples, let's say many real examples. And so if we throw away the color, just discarding the y variable, we're left with something you can call the obscure genealogy. So it's got the topological structure, but not the color. So what I want to do is I want to show you what the results look like for the likelihood both of a prune genealogy and of an obscure genealogy. So this is a 15-minute talk, and I neglected to look at the thing, so you'll tell me. But this is a no-win situation, right? I could either spend 45 minutes giving you enough background so where you could understand the notation before I state the theorem, or I can state the theorem with the notation and you won't understand it, or I can just give you something compact so you can take away the. So yeah, so the question is, I won't mess with that because that's dangerous. So we're going to model the sampling process just like we model anything else. We can model it either in terms of an effort of sampling that's distributed through time, batch sampling events where we sample a whole bunch of individuals, any combination of those things. We can condition on the samples, we can not condition on the samples. All that really from the mathematical point of view we have to have is something that determines that genealogy, right? So these are, the simplest way to think of it is they're samples of the genome of, say, a virus that were taken from an individual at a known time. The number of samples is part of the data. It's not part of the, I mean, it's part of the data. Okay. All right. So what I opted to do in face of this no-win situation was to show you the form of the equations and tell you a little bit about what they are in the hopes that we can chat more about it if you're interested. So this is a nonlinear filter equation for a quantity W. You'll notice that it bears a striking resemblance, the first equation bears a striking resemblance to the Kolmogorov-Forward equation, and indeed the Kolmogorov-Forward equation is a filter equation. If we just set this phi term to 1, then we're left with the Kolmogorov equation. This phi term, of course, therefore, is the really interesting thing, and that is a number between 0 and 1. It's actually a ratio of products of binomial fractions, binomial coefficients, that is completely determined, the form of which is completely determined by the model. So given a model, we can work out what those phis are once and for all. I won't bore you with the details of exactly what that ratio looks like. If you're interested, I can show it to you. It's not that terrible. And it's a function of the state of the system and the local color structure. The local color structure at the point Y, at the point T on the tree. It's got a regular portion that holds, this first equation, that holds at every point on the genealogy, at every time, that's not the time of an event on the genealogy, not the time of a sample or branching point on the genealogy. And then a singular portion which holds at all of those events. This tilde notation I use to represent, the left limit of a CAD-like function, for those of you that care about this. This is basically just looking at the same point in a discontinuous function from the left or from the right. So the tilde represents looking at it from the left. And then we have the theorem that tells us that the likelihood is the sum of this density W across the entire space at the final time. So we start this thing off at the initial time, and if we can solve this equation from forward in time, then we simply sum it up at the end. So yeah, that's what I meant to say here, is that we can solve this from left to right. There's, of course, also an adjoint form to this, in the same way that the Kolmogorov backward equation is adjoint to the Kolmogorov forward. And it looks like this. If we have the final condition, which is just that this function is equal to 1, then solving this adjoint form of the equation, where the terms are all the same, it's just been slightly rearranged, then if we take if we integrate this function at time 0 against the initial density, we get the likelihood that way as well. Okay, so this is for a prune genealogy, that is a genealogy that doesn't have individuals we can't see, only samples, but does have the colors, does have the Dean locations of all of the lineages. And of course, in many, many important cases, we won't have that. But there are some cases where we will have that, actually. For example, if we've got 2 strains or 2 or more strains of a pathogen that we know a priori are distinct from one another, we know that they diverged in the distant past before our period that we're focusing on, then we know a priori that what the colors of the trees are. And there are, of course, other situations as well. And one can, so sometimes use this theorem directly. That's what I was going to say right there. It also covers the 2 special cases that are at the foundations of all the existing widespread phylodynamic methods, both the linear birth death and unstructured coalescent processes. It's fairly straightforward, although non-trivial in the case of the linear birth death process to demonstrate that. Okay, so what we want to do now is move on to working out what the likelihood of an obscure genealogy is. Okay, so the idea here is that we're going to have to integrate over, what's that?
We gave you extra technical minutes, but still.
Stoppage time, huh? All right. So this theorem is quite a bit more complex looking, but it's really the same idea. The only thing is that we've augmented the state of the system with the additional variables y to represent the color. Okay, so both the forward and the adjoint forms of this equation are very much the same. Again, these boost functions are once and for all determined by the form of the model. Okay, so just to go back to the simple case and show you what the filter equations look like for an SIRS model. This is a 1-Dean situation, so the first theorem applies here. There's no need to consider the color because there's only one color. And here's what the filter equation looks like. It's a little bit more complicated. You can see a couple of expressions here. And those of you that have worked in this area will recognize this as being very, very close, in fact, to approaches that have been proposed in literature before. There's some complexities here that haven't been noticed. And one can compute with this effectively. That's not too complicated. This is a little bit more of a test of things. We've now got the fully structured problem. And I couldn't fit the filter equation. I could almost, but not quite fit it on the page here, so I won't bother to show that. But for these filter equations, there's always a naive Monte Carlo algorithm that one can apply. So it's sort of determined by the model and nothing else. It's not guaranteed to work very well. In this case, it works well enough to be able to see that there's information in the data about the value of this parameter. So I stop there before more technical things happen. Just to remind you that this theorem really corrects and strictly extends all the existing coalescent phylodynamic methods into a single framework. Of course, there's more that can be done, but I think it's quite interesting to ask what can we do with this formalism. And the key thing is that, at least in many cases, or if one adopts a sufficiently expansive notion of what it means to carry out a computation, then this massively expands the class of models that can be entertained. So there's things to do still. I'd love to talk with you about them. We're going to have a discussion this afternoon at the first session after lunch. And one of the topics we want to think about, I think, it's quite open, but one of the topics that we can think about is additional areas beyond epidemiology where these kinds of approaches might be useful. And we've already seen some today. All right, so I'll stop there. Thank you.